Have you ever wondered how life began on Earth? For decades, scientists have been trying to piece together the earliest moments of life, when the very first molecules began to replicate and evolve. Now, an exciting breakthrough brings us one step closer to understanding that ancient mystery. A team of researchers has successfully recreated a key process that may have triggered life's origin billions of years ago, by getting RNA to partially self-replicate under early Earth-like conditions. This could be the closest we've ever come to replicating the spark of life itself. In a recent study published in Nature, scientists from University College London and the MRC Laboratory of Molecular Biology in Cambridge have demonstrated that RNA molecules can begin to replicate themselves, even without complex enzymes. They used simple chemical components called RNA triplets, short three-letter sequences of RNA, to recreate conditions similar to those found on the early Earth. These triplets acted like basic building blocks, forming the foundation of primitive genetic material. But here's the twist: the team didn't use a warm tropical soup. Instead, they recreated icy, acidic freshwater environments, something you might find in a geothermal pond surrounded by glaciers. These cold conditions actually help the RNA strands replicate more efficiently. Why? Because freezing slows down molecular movement, preventing RNA from forming its double helix shape too quickly. This delay gives RNA the chance to copy itself before locking back into a stable structure. This research supports a fascinating theory known as the RNA World hypothesis, the idea that before DNA and proteins, life began with RNA. RNA is unique. It can store genetic information like DNA, but it can also act like a protein, serving as a catalyst for chemical reactions. In a way, it's the Swiss Army knife of early biology. For life to begin, molecules had to copy themselves, reliably and repeatedly. Until now, scientists weren't sure how this could happen naturally, without lab-made enzymes. But this new study shows that RNA can, under the right conditions, partially replicate itself, naturally. In the experiment, the scientists achieved 17% replication. That might sound small, but in molecular biology, that's huge. It means we're closer than ever to understanding how life might have started from nothing more than a few simple molecules. So why is this discovery so important? For one, it bridges a gap between chemistry and biology. We now have a clearer picture of how non-living molecules could become self-replicating systems, the first major leap toward life. Secondly, it opens the door to recreating these early life steps in the lab. If scientists can refine the conditions to improve replication efficiency, we might soon witness the full, enzyme-free formation of a replicating RNA system, the kind of process that led to all life on Earth. And there's another exciting angle: astrobiology. If RNA can replicate under icy, acidic conditions, then similar processes might be possible elsewhere in our solar system. Think Europa, Enceladus, or even ancient Mars, icy worlds with geothermal activity. This research doesn't just tell us about our past, it could help us discover life beyond Earth. In summary, scientists have just made a key discovery: partial self-replication of RNA in early Earth-like conditions. It's not yet the full recipe for life, but it's a giant leap toward uncovering the molecular origins of existence. For the first time, we have experimental evidence that supports how life might have started: cold, simple, and beautifully chaotic. As research continues, we may soon witness a lab-based recreation of life's first spark. From there, who knows what mysteries we'll unlock about our origins, our planet, and our place in the universe. If you're fascinated by the mysteries of life and space, hit the like button, subscribe for more science updates, and share your thoughts in the comments.